Okay, last set of notes here on organic chemistry, and these, unfortunately, there's a lot of memorization on the front end here. I'm going to go through the back part of this pretty quickly because there's not a lot of that sort of thing on the AP exam, which is unfortunate because it really is interesting stuff, and it's the kind of stuff that I think would be fun to teach in an organic chemistry class. Um, so anyway, we're going to go through this fairly quickly. But when we talked about functional groups, what we've been talking about so far are just straight carbon chains. So carbon single bonded to another carbon, and then hydrogens connected to that, okay? Or maybe a carbon-carbon double bond like this, or triple bond. That's all we've talked about so far. And really, as far as organic molecules go, there are some of those in the body. And those kind of form the backbone of everything else in the body, okay? More of what you find in the body is going to have what's called functional groups on it, and that's just things that are attached to these carbon-hydrogen chains that make them a little bit different, okay? And so we're going to go through each of these, well, not each of these individually, but a lot of these different groups we will talk about. Let's see here. The ones that we've already talked about, we've talked about the alkenes, okay? That's just a carbon-carbon double bond. And the alkynes, that's the carbon-carbon triple bond. The other one that I don't think that we actually go over in any detail is the haloalkanes right here. But that's fairly simple. That's just you've got a carbon-carbon chain and then you have some sort of halogen attached to it. That's haloalkane. So it could be a chlorine, it could be a bromine, any of the halogens, fluorine, iodine. When you have those attached to a carbon, it's called a haloalkane. Okay, and we're not going to talk a lot about how to actually name these functional groups because what I've seen on the AP exam emphasizes just recognizing the functional groups, not naming them. Okay. So I think that's probably all you're going to be responsible for. Again, I would love to take more time on this. We just don't have the time. So we're going to go through this pretty quick, talk about what you're looking for with each of the different functional groups, and I'll give you some things to help you identify each of these things, and then we'll go from there. So this up here. First group we want to talk about is the alcohol group, okay? And these are called hydroxyl groups, which is just an OH group, okay? So anytime you've got a carbon-carbon chain with an OH group attached to it like this, that's what defines alcohols, okay? So anytime we call something an alcohol in organic chemistry, we're referring to something that has an OH group on it. And these are usually pretty easy to identify, okay? Sometimes they're not completely obvious, just depending on the way that they're written structurally, but you can usually tell, okay, this has 3 carbons, and then on the end of it we have an OH group, okay? And so then here's kind of the way that these are named. So if you've got the OH group off of there, if this is your, it doesn't matter which way you number from first, second, third carbon here, then we're going to call this propanol. You put the ol ending on it to show that this is an alcohol, okay, and this is 2. But again, typically we don't talk about the naming on these. The alcohols are a little more common, so I thought maybe I would mention that a little bit. But you can see this other one over here is 2-methyl-2-propanol because we've got a methyl group up here, and then we've got an alcohol group down here. And then if you have 2 alcohol groups, it's called diol because di is the prefix for 2 of them, okay? And then you can see each of these has more common names, but this is the naming system that is most systematic, that makes the most sense across the board. So that's alcohols, okay? And we just talked about all of that. So. Alcohols are more acidic than hydrocarbons, okay? And we haven't really talked a lot about the acid-base, the pKa stuff yet, so we'll get to that later. But what that basically means is if you've got something like this, carbon, 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 oxygen, hydrogen, okay? This oxygen right here is pulling electrons towards itself a lot stronger than any of the carbons is pulling electrons towards itself that's attached to any hydrogens over here, okay? So if you have just a normal carbon-hydrogen molecule, that's not very acidic because these hydrogens are sharing fairly equally with the carbons. They don't really want to leave. But if you've got an OH group, this hydrogen right here barely is holding on to the electrons at all because the oxygen is pulling so tightly on them. So it's very easy for this hydrogen to get pulled off when put into water and increase the H plus concentration in a solution. Okay, so that's why they're more acidic. Okay, next group, ethers. And ethers basically just have an oxygen in the middle of the chain. Okay, so kind of different there. What you're looking for with those, you're just looking for something where an oxygen is bonded to two different carbons. Okay, so you've got this central oxygen here. Ethers are anesthetics. They tend to put people to sleep when people breathe them. And so that's something that's used a lot in medicine, obviously. And we still don't quite understand exactly why those work. I had my son got put to sleep this summer because he was having a surgery on his throat, and they had to put him to sleep for that. And it was funny because the anesthesiologist came and talked to him, and my son was asking him all sorts of questions like, you know, why does this work, you know, that sort of thing when they were talking about putting him to sleep. And the anesthesiologist said, well, scientifically, we still don't really know exactly why it works. So I'm like, well, that's a comforting thing for a parent to hear right before his son gets put to sleep. But anyway, he was fine. But these tend to be unreactive because if you look at it, there's a little bit of polarity there, okay? But because the oxygen is in the middle here, there's not really a lot that can react. So they're good polar solvents, okay? So they have this polarity because you've got, if you think about it, you've got the two lone pairs on here that are going to, you know, change the shape of this thing. But because they're fairly unreactive, you can dissolve stuff in it, like dissolving stuff in water. So if for some reason you don't want to use water as your polar solvent, this, you know, ethers can take that place. So again, the main thing you're looking for here, you're looking for this oxygen in the middle, okay? Carbonyl compounds. There's a lot of different carbonyl compounds, okay? And so if you hear that term, basically what that means is there's a carbon-oxygen double bond, okay? So you notice in all of these different categories here, the amides, the aldehydes, carboxylic acids, the esters, and the ketones, you have carbon-oxygen double bonds. So what determines whether it's, you know, which one of these five it is what else is attached to the carbon that's double bonded to the oxygen. So notice here on the amide, you've got a nitrogen attached to the carbon, okay? And this R right here, that just means, you know, you could have like 4 carbons before you get to this. Okay, so this right here is just, it kind of means the rest of the molecule. That's not what R stands for, but I guess you could think of it that way. R equals rest of molecule. So the amide, again, has a nitrogen attached to the carbon. The aldehydes, you're going to have a hydrogen attached to the carbon. Okay. Carboxylic acid, you've got an OH group, so it's sort of like an alcohol. And again, it's called an acid because this H right here, it's very easy to break this bond, and this H can be donated in solution, increasing H plus concentration. An ester, you've got another oxygen here. Now, these are hard to tell apart, okay, the carboxylic acids and the esters, because they both have oxygens, but what you have to remember here, the carboxylic acid, it's going to donate a hydrogen. So think about the acid part, all right? There needs to be a hydrogen that can be given away. On an ester, there's just, you know, more carbons on the other end. So this is right in the middle of the chain, okay? And this oxygen right here is going to be attached to another carbon. And then ketone, we just got carbons on either end, okay? And so, you know, some of these are a little bit hard to tell the difference, but because they're very similar, but you just have to pay attention to what's attached to what, and that should help you out on these. It's just going to take some studying and some memorization, unfortunately. Okay, so the aldehydes, again, at least one hydrogen attached to the carbonyl carbon. Now, when we say carbonyl carbon, we mean this one, the one that has the carbon-oxygen double bond. That's the carbonyl carbon. So you've got a hydrogen there, that makes this an aldehyde. Okay? If you got a ketone, then again, you can see how this kind of works. The carbonyl carbon right here, it's attached to carbons on both sides. Okay? So that's what defines a ketone. Carboxylic acid. Here's the carbonyl carbon, and you've got an OH group attached to it. Okay? So again, this H right here, that's what's going to signal you, oh yeah, this is the one that's the acid, the carboxylic acid. Okay. These carboxylic acids are tart tasting, and they're weak acids. Okay. And so an example of that you're very familiar with, CH3COOH, that's acetic acid. Okay, acetic acid, the actual structure of it looks like this. So you've got a carbon double bonded to an oxygen, and you've got the OH group on the end. And so the reason that this is an acid is because of this hydrogen right here. Okay, and but these are weak acids, so they don't completely dissociate in water. They partially dissociate. But acetic acid is a good example of one of these carboxylic acids. Here are some other examples. Citric acid, okay, very familiar with that. Lactic acid. Formic acid. Formic acid is actually the acid that ants make when you get ant bites. And so those are some of the ones. Acetylsalicylic is aspirin, so you're probably very familiar with that one as well. So a lot of different types of carboxylic acids are very important. We experience them on a regular basis. And you can see what the structure of all of those are. But, I mean, ultimately, you've got these carbonyl carbons that have OHs coming off of them, okay? And so that's what's going to define a carboxylic acid. Hopefully that makes sense. Esters. Okay, esters are actually important in fragrances, and these get produced because of a reaction between a carboxylic acid and an alcohol, okay? And so when that happens, you end up with these, well, essentially what happens here, this did have a hydrogen attached to it before, okay? That hydrogen comes off, and then you end up with this, excuse me. So this was the carboxylic acid here, okay? Sorry, this part right here. And then this was the alcohol, okay? And the alcohol gives away its OH group, and the carboxylic acid gives off its hydrogen. That just got messy. You just imagine a hydrogen being here initially and an OH being here initially, okay? Those come off, these two things come together, and then you end up with an ester, okay? And esters, we're actually going to do our lab on esters next week. And a lot of good-smelling fragrances are produced using esters, especially things that have like fruity smells to them. And so we're gonna produce some of those in the lab by doing some of these reactions between acids and alcohols. And we'll make some of our own esters and see what the scents are and see if we can figure out what they smell like. So that should be kind of a fun lab. Again, found in many fruits and perfumes. Okay. Amides, or amides, that's going to be, again, on your carbonyl carbon, you're going to have a nitrogen attached to it, okay? And these are formed by the reaction of carboxylic acids with amines. Now, we still have to talk about what amines are. But amides, you're going to have this carbon-oxygen double bond, and then you have a nitrogen attached to it, okay? Amines, you just have a nitrogen attached to a carbon, okay? And amine might sound familiar in the sense of like amino acid, okay? And so these are sort of the base of amino acids. Whoops, let me go back. These typically have strong unpleasant odors. Okay, and so again, what you've got here, notice there is no carbon-oxygen double bond on the amines. It's just carbon, carbon, nitrogen. Okay, and there's a couple hydrogens attached to that. So that's the way these structures for amines work. If it's an amide, there's going to be a double bond here, carbon-oxygen double bond. Okay, so that's the difference between amines and amides. Now, amino acids, what you're looking at here is you've got an amino group and a carboxyl group, okay? That's what makes the acid part. So you've got your amine group, and notice here you've got this carbon single bonded to a nitrogen here, and then you also have a carbonyl carbon connected to a nitrogen. So we've kind of got an amine and an amide in the same thing. And then you've got another, amide there. And then you've got your carboxylic acid on the end, okay? And this one has already given away its hydrogen, but there was a hydrogen there. So that's the idea with these amino acids. It's a combination of an amide and an amine and a carboxyl group, or a carboxylic acid, okay? Proteins are a bunch of amino acids put together. I know you guys learned this in biology, but it's been a couple years. So there's an amine end on one, and there's an acid end on the other. Okay, so you've got this amine end here that can connect to the acid end on another amino acid, and you can just string these together in long chains, and this makes proteins. Okay, and this is called a peptide bond. Okay, and if you get a peptide bond in a certain sequence, then you've made a certain protein. Okay, so these are obviously very important in the body. Okay, and so you got these peptide chains, and what they start to do is they start to loop around themselves, and they have these. You have these carboxylic acid groups on the ends of these things, and then you've got the amine groups on the other ends. And you've got a nitrogen-hydrogen bond here, and then you've got a lone pair on this oxygen. And so these are attracted to one another, this hydrogen and this oxygen. And so what it does is it starts to pull this thing into a helical structure, sort of like with DNA. And so these things wrap up, and they coil, and then you end up with a protein. And that's what gives them their particular shape. If you've ever heard of denaturing a protein, denaturing an enzyme. Enzymes are specific types of proteins that speed up chemical reactions in the body. What happens there, if the protein, if you change the temperature too much, you change the acidity that the protein is in, it actually does end up breaking some of these bonds, and it changes the shape. And once the shape of the protein changes, it can no longer function the way that it's supposed to anymore. So one of the things that actually happens when you freeze to death is that your enzymes start to denature, and they can no longer speed up the chemical reactions in your body the way that they're supposed to. And so your body just kind of shuts down. And so that's, if you get exposed to extremes in temperature, or if your body somehow becomes more acidic than it's supposed to, or more basic than it's supposed to, then it can actually change the folds of these proteins, and then they don't work correctly anymore, and then your body starts to shut down. Most enzymes are proteins, so I don't know if you talked a lot in biology about the lock and key model. If any of you are in anatomy and physiology now, or maybe even AP Bio, I'm sure you're talking about this sort of thing, or you will at some point. So basically, the way that these enzymes work is they provide docking stations for these two chemicals to come together. And so the shape of the enzyme is very important, because if the shape is not right, then these things can't dock where they're supposed to, and they can't do the reactions they're supposed to. And most enzymes are very specific to one particular reaction. So we have an enzyme in our body that is specifically for breaking down hydrogen peroxide. Our body, through some of the chemical reactions it does, makes hydrogen peroxide as a byproduct, and that's very toxic to our bodies. So if you put hydrogen peroxide on a cut, that enzyme is going to break that hydrogen peroxide down. You've got an enzyme in your skin that does that, and it breaks it apart into water and oxygen, and the oxygen is actually very healing for that cut. But if you have that inside your body, a high concentration of hydrogen peroxide that's very poisonous to our bodies. So that enzyme immediately starts to break that hydrogen peroxide apart into water and oxygen, which are harmless to us. So that enzyme is very important. If that enzyme's not working, then we die of hydrogen peroxide poisoning. Okay. Carbohydrates, just real quick about those. So again, going back to this, I guess one thing that I want to emphasize here with these amino acids is just noticing, because I think this is one of the questions on your AP. Well, here, I can show it here, I guess. Notice the elements that are present in these amino acids, these proteins. You've got carbons, you've got hydrogens, you've got oxygens, and you've got nitrogens, okay? And those are the main components of proteins, and of DNA for that matter. Okay, so we went through this already. Let's talk about carbohydrates, okay? Carbohydrates, these are going to be aldehydes or ketones, typically, okay? And so we're talking about things that have carbon, hydrogen, and oxygen in them, all right? And these are long, long chains of carbon, hydrogen, oxygen, okay? And so these carbohydrates, these sugars, they're going to be a little bit polar, okay, because of the oxygen here. So this is an aldehyde, notice, because it's got the hydrogen attached to the carbonyl group. And then this is going to be a ketone because it's got carbons attached to either end. And so this is an example of glucose here. This is an example of fructose. And so you can see these sugars are just these long chains of carbons with these carbonyl groups attached to them at some point. When you put these into solution, they actually form these cyclical structures. They sort of cycle up a little bit, and so you end up with these rings of carbon and oxygen. Let's see if there's anything else here. These cyclical structures, they can form chains here, and they make things like starch, cellulose. Cellulose is the compounds in trees and other plants that give them that really tough, sort of hard to chew, and our bodies can't really digest them. And then you've got starches, and we use starches for a variety of different things, for cooking, for even people used to use it for clothes. I don't know if people really use it for that anymore, but it makes your shirts kind of stiff, and I guess they look newer, that sort of thing. So anyway, important things. And then nucleic acids. Again, now we're back to the carbon, hydrogen, oxygen, and nitrogen. Okay, and the way that these work is they've got three different groups on them. Okay, so and you learned this when you learned about DNA. You learned about the three different parts of the DNA molecule. One of them is a sugar, ribose or deoxyribose. For DNA, it's deoxyribose, obviously, and for RNA, it's ribose. And then you've got your bases, the adenine, guanine, cytosine, thymine, or uracil, and this shows the structure of all of these down here. Okay, lots of nitrogen in those bases, so we've got a lot of carbon-nitrogen bonds here, and then some oxygen as well with the guanine and the thymine and uracil. Well, actually, yes, cytosine too. The only one that doesn't have the oxygen bond is the adenine. And then the third part of the DNA, besides the sugar, the base, the A, C, T, G, or U, is the phosphoric acid group. Okay, you got this phosphate on there. And so these are really the three different groups on the DNA. And so, again, chemically, you can sort of see how these organic molecules just become incredibly important to living organisms, because this is what makes up the DNA molecules. So you've got these, all of these bonds here. There's a, excuse me, this is an alcohol right here. You've got this carbon connected to an oxygen connected to another carbon here. So we've got all these different functional groups that are working on these carbons. You've got, you know, amides and amines up here on the adenine, and then you've got your phosphoric acid group over on the side here, and that forms what's called a nucleotide. Okay, and the nucleotides, that's what's going to form that helical structure, that double helix that we're used to with DNA. Okay, and so again, there's not a lot on this organic, the last part of this, the biochemistry stuff on the AP exam, but they do ask questions about it every once in a while. They want you to know the composition of nucleic acids, carbohydrates, DNA, that sort of thing. So it's good for you to just sort of be reminded of those things, although you may see one question on the AP exam. The thing that you really need to know from what we talked about today is you need to know the functional groups, okay? And so I'm going to go back. What I would do is I would look at this chart at the beginning here. Oops, too far. I would really pay attention to this chart, okay? Because this chart, it shows you what the functional group looks like. It gives you an example of each of these things, and you need to know these, because this is the question I've seen more often than not on the AP exam is, you know, which functional group is included on this organic molecule, and then you have a multiple choice question there where you have to pick which one it is. And so you need to know these, okay? So anyway, that's organic chemistry in a very, very short time period. I wish we had more time to spend on that because it really is very interesting stuff, especially for those of you who are interested in medicine or pharmacy or anything like that. Very, very important things for you guys to know. So anyway, that's that, and that's all the new stuff we need to cover for this unit. So we'll do our ester lab on Tuesday, and we'll take our test. No, I'm sorry, we'll do our ester lab on Wednesday when you guys get back, and then we'll take our test on Friday.